Hello students, continuing with a series of lectures, Biology Made Easy. This lecture is about microbiology. Microbiology is a branch of biology which deals with the study of microorganisms. Microorganisms are the organisms which are very small. They are called microscopic. They are so small that they are invisible to naked eye. An instrument called microscope is required to view them. The fact that they are small that makes beginners to think that they are difficult to understand, that microbiology is a difficult subject. But believe me, it's not that much difficult. The subject microbiology requires a clarity of concepts and an imagination also in order to understand. In my lectures, I will try to explain the subject in a simple manner. The microorganisms are present everywhere. Actually, we are living in an ocean of microorganisms. And it is a belief that if there is a microorganism, then it will cause disease. Only 5% of microorganisms are pathogenic. That means only 5% can cause the disease. Rest of them are not harmful, and a large number of them are beneficial for us. We can utilize this microorganism for our benefit also. I am going to explain the microbiology with a simple experiment, the conversion of milk to curd. Curd is a healthy dairy product. It is considered as superfood. It is much more healthier than the milk. We all consume curd, and every kitchen is involved in the routine preparation of curd. Curd is almost prepared daily in the households, and we don't know that it involves the microorganisms. We are carrying out this process almost daily without knowing that we are carrying out a fermentation process. Fermentation process in which milk is converted to the curd. The friendly bacteria lactobacillus is involved in this conversion. It changes the sugar of the milk to the acid. The acid which is produced, that increases the acidity of the milk, which is responsible for the curdling of the milk, so that the curd is formed. The sugar of the milk is lactose. It gets converted to the acid, lactic acid, by number of species of lactobacillus. What is the procedure of the curd formation? In the first step, first of all, the milk is boiled to the boiling point, and then it is let to cool down around 45 to 50 degrees Celsius. Actually, in the house we are not monitoring this temperature, but by touching, by feeling, we get to know that the milk has now cooled down. Now the starter culture, actually it is called starter curd, is added. We add a portion of the curd from a previous lot. It is used as a starter. The starter is actually containing the bacteria. It is then mixed and covered and kept at a warm place. The curd sets in 3 to 6 hours depending upon the room temperature and the climatic condition. Now that the curd is all set, how will we get to know that it contains bacteria? We need to observe the bacteria in the curd. For this purpose, take 2 to 3 drops of the curd on a clean grease free glass slide. Spread it using a loop so as to make a smear. Let the smear to dry in air. Now add a drop of methylene blue for staining purpose. Some other stains can also be used. Keep it for 1 minute. Wash it off with tap water and blot it dry. Now examine the slide under high power objective. High power objective is the 100x objective lens of the microscope. What is this objective? What is the high power? That we will discuss in coming lectures on microbiology. This shows the examination of the slide. The slide which we have prepared, when it is observed under the microscope, it shows the presence of mixed bacteria. Mixed bacteria means not only a single type, but there are different types. Out of those different types, the most common is lactobacillus. Other bacteria like streptococcus, lactococcus, leuconostoc or yeast can also be seen. This shows the lactobacillus which are rod shaped, streptococcus which are spherical shaped. One more product is yogurt. It is also a dairy product, fermentation product prepared from milk. Yogurt, yoghurt or yoghurt. It is the same product named by different names. Nowadays, it is becoming very popular as a diet food. It is a probiotic food which contains lot of good bacteria which are required for the good intestinal health of the human body. It is a prevalent myth that the same product is called curd or dahi in India and it is called yogurt in the West. But these are the different products. What is the difference? The difference is that the yogurt contains standard strains of Lactobacillus acidophilus and Streptococcus thermophilus. These are added to the milk which is already pasteurized. Then temperature is maintained for 4 to 12 hours so that the fermentation can be carried out by the bacteria. The curd is different because in the curd we are adding a starter curd, and its starter curd also contains the bacteria, but those bacteria are not the standard strains. So that is the difference between the yogurt and the curd. I hope this lecture will help you in studying of the microbiology. If you have some queries, suggestions, please feel to contact at this email ID. I will be glad to hear from you. Thank you.